So we're going to be talking about changes in the DNA, and changes in the DNA are called mutations. Mutations, one of the keys is that they have to be heritable. That means they have to be able to be passed on to offspring, and not all changes in our DNA can be passed on. The ones that can be passed on are going to be changes in the bases, so in the nucleotides. So for example, if you have an A in the nucleotide and that becomes changed to a G, that's the kind of change that you can pass on to other cells in your body or to your offspring. Mutations often get a bad rap, but they actually do have a very positive side. The mutations account for all of the variability among organisms. So mutations are the cause of the different traits in humans, and they're also the cause of the different traits in species. And so we would have never gotten to the Earth that we have today with the millions of species that are present on the Earth if mutations didn't occur. But on the other hand, most mutations are harmful rather than beneficial. And so for that reason, our bodies and our cells have repair systems that will fix changes in the DNA and repair that DNA damage. And so we're going to talk about the different types of mutations, some causes of mutations, and then briefly talk about the repair processes in the cell. So a point mutation is a single change in a base pair. The first kind is the base substitution, which is simply going to change a base. So for example, your original sequence might be a double helix like this. And remember, it's a double helix, so the other side is this part. And the mutation might change this A into a G. But remember, because of base pairing, this will eventually also become a C. So that's why we say it's a single base pair change. And again, this is a base substitution. I substituted a GC base pair for an AT base pair. Additions and deletions are the adding or removing of a base pair. So the best way to look for these when you're looking at sequences is to make sure you make groups of 3. So if I go ahead and divide the before and after sequences into groups of 3, oops. Oh, sorry, I'm doing that on the wrong one. I was very confused for a second. Okay. So if I go ahead and do this on the before and after sequences, you'll notice something really important. On this original sequence, there's only 2 after I do groups of 3. But on this one, there's now 3. So that means that in this sequence, there's a total of 11 bases. And in this one, there's now a total of 12 bases. So pretty simple to see that this is an insertion. So there's an extra base. Now I need to look for where that insertion happened. And of course, this is highlighted for you, but imagine you're trying to find this for yourself. You look at your groups of 3. So my first group of 3 is the same. But if I look at my next group of 3, GCT, it's different from this one. So this must be the group of 3 in which it originates. And so then you want to look for that GCT if it's still there. And so, oh, there it is. That GCT is still there, it's just not in my original group of 3. And then if I look down, there's AGA, and there's my original AGA, and then there's my TC. So that points out that this must be the base that was added. So this is an addition or insertion of an A into the sequence. Insertions and deletions also do something else. And so remember that when we were talking about a translation, we talked about the importance of the start codon in establishing the reading frame. And so I'm going to use this sentence again. Oops. The cat ate the rat. And as long as I group like this sentence makes sense. But and let me rewrite the sentence here. If I change the grouping, so in this case I added a nucleotide, I added a T, that extra base causes my groupings to be different. And so all the way down, now it says th, eka, tat, eth, ura, t. And so now it doesn't make sense anymore. It's not the same thing that this said. all because I changed the grouping for the whole sequence, even though I only added this one base. It made everything after that addition be messed up. This is called a frame shift mutation. And frame shift mutations are caused by additions and deletions. So the mutations we've just been talking about answer the question of how does the mutation change the DNA? So changes to the DNA are described with the terms, again, substitution, a base substitution, addition, deletion, and frame shifts. All of these terms describe the changes that happen in the DNA, but the DNA is going to be read to make protein. So the second answer about mutations is answering the question, how does the resulting protein change? So how does that change in the DNA lead to changes in the protein? And those can either be silent, they can be a missense, they can be nonsense. And we're going to go through what these terms mean. So a silent mutation is one that doesn't change the amino acid sequence. So silent means no change. This is going to be particularly common in sequences, in mutations that happen in the third position. So remember that if you have, say, a TAT, and the mRNA is going to be an AUA, so this is the DNA, this is the mRNA codon. In the genetic code, this third position could be more variable and still get you the same amino acid. So go back and look at that genetic code, and again, remember that, say, serine had 4 different codons that all led to serine, and they were different only in this third position. So if you get a difference in the third position, you're more likely to have a silent mutation. A missense mutation is when an amino acid is changed. So for example, maybe you used to have a serine, but now you have a proline. And so again, this is going to be the most common result of a substitution. This can have really significant effects on an individual, or it cannot affect an individual very much. It depends on the amino acids involved. Remember that some amino acids are polar, some are nonpolar. So sometimes if you substitute a polar amino acid for another polar amino acid, you won't get that big of a problem. But if you substitute a polar amino acid for a nonpolar amino acid, you might have a really significant effect on the person's health. And so sickle cell anemia is a disease that's caused by a single substitution. So it's a substitution in the DNA that leads to a missense. So again, when you describe mutations, you should ask yourself what happened in the DNA and what happened in the amino acid. So this is the a part of the hemoglobin protein. It's the beta globin gene. And so you can see that this is the substitution. We used to have an A in that position. Now we have a T in that position. So that will code, the normal sequence codes for a glutamine, which is a polar amino acid. The new mutated sequence is for valine, which is nonpolar. And what that causes is a place in the protein that's nonpolar that shouldn't be. And then that causes the hemoglobin fibers to clump together in a way that they don't normally clump together. And you can see these hemoglobin fibers here that don't normally form. And when they form these long fibers through the red blood cell, they pull the red blood cell into this curved shape. A normal red blood cell looks more like a filled donut. But the sickle cell trait will cause the red blood cells to have the curved shape, which is where the name sickled comes from, or sickle cell comes from, is for a curve of a sickle. And so again, this is a single base substitution, but it leads to a single change in the amino acid, so a glutamine to a valine, and that is going to change the whole structure of the protein that this is part of. and then that affects the shape of the cell. So again, even though it's one small change, it can have profound health consequences. Nonsense mutations are changes that will change a regular codon, so an amino acid, in that position into a stop. So it ends the protein early. So it's going to cause protein synthesis to end early. So you will end up with a shortened protein. The rest of the protein won't be translated. So once you see the stop codon, no other amino acids are added on to that. And then frameshift mutations we talked about already. These are going to create, these are because of additions and deletions. Sorry, let's remind us that it's additions and deletions that cause the frameshift. The additions and deletions cause the frameshift, and then frameshift mutations will cause something called extensive missense. So you don't just get one amino acid sequence that's wrong, you get multiple amino acid sequences that are, or you get the whole sequence that's wrong because the reading frame is completely altered. So extensive missense means many amino acids are different. So this is a summary of the types of mutations. This figure is on D2L if you want to print it out. So you can have a base pair substitution. This is a U instead of a C, and in this case, this one is silent. So this is still coding for a glycine. So the original sequence coded for a glycine, this one still codes for a glycine. This is a different substitution, so it's still a substitution, an A instead of a G, but this one is now a missense, because instead of a glycine, like I originally had, I now have a serine. This is a base substitution, a U instead of an A, but now it creates a stop codon early, so this protein is going to stop being made, so none of this will get added on. So this is a nonsense mutation. These are examples of insertions and deletions. So in this case, there's an insertion of a U, and in that case, it creates a nonsense mutation. So sometimes an insertion or a deletion will create a nonsense. This is a frame shift from a deletion. So here's a deletion of a U, and now I'm getting all of my groupings off now because of that. And so all of the amino acids that follow are going to be different. So this is that extensive missense. This one is kind of interesting because instead of just a point mutation of one nucleotide, it's a deletion of 3 nucleotides. And what you notice is that causes this lysine to be missing, but it does not cause a frame shift because all of these will still be grouped correctly because it was a deletion of 3 nucleotides. So that's just kind of interesting. Deletions in multiples don't always cause a frame shift. So the last differentiation of mutations that we'll talk about is the difference between a germline mutation and a somatic cell mutation. Germline cells are the cells that will give rise to sperm and egg. So they're a very special set of cells in your body. They're reproductive cells that will become or produce eggs and sperm. Somatic cells are basically all the rest of the cells in your body. So these are your body cells, cells that will not contribute to eggs and sperm. These are significant differentiations from mutations because whether mutation happens in the germline or somatic cell is going to affect whether it can be passed on. So if a germline mutation occurs, and so in this case it's happening in the sperm. So a new mutation in a sperm, the sperm fertilizes the egg, now the whole embryo has the mutation. So every single cell in the embryo has the mutation. So every single cell in the adult organism will have the mutation. So germline mutations will end up in the whole organism, and then because he has it in his whole organism, he can pass it on to his children. So that his sperm that he makes can also carry the mutation. Not all of them will carry the mutation. So just because you have a mutation doesn't mean you'll always pass it on, but some of his children can get the mutation. A somatic cell mutation happens after the sperm and egg. So in this case, it's happening in the embryo. So the sperm fertilizes the egg, both were not mutated. So your embryo mostly doesn't have the mutation, but sometime during development, a little patch of the embryo gets a mutation. So in the adult organism, there's going to be just a part of the organism that has a mutation. So in this case, it's his shoulder, right? Well, his shoulder has nothing to do with making sperm. So when he goes to make sperm, he's not going to be able to pass on this mutation to his children. So none of his children will get this. So the other place where somatic mutations are really significant, or the disease in which they're significant, is cancer. Cancer is almost always the result of somatic cell mutations only. So somatic cell mutations will occur over time, someplace in the body. If a lot of those mutations accumulate, then that can create cancerous tissue. But those cancerous cells with the mutations are not going to be passed on to the offspring because they don't contribute to egg and sperm. Now, some of you maybe have heard of inherited cancers, and this is perhaps most famous to most people in breast cancer. There's a particular inherited gene called the BRCA gene, which stands for breast cancer gene. And so BRCA is a mutation that some people have that predisposes you to breast cancer. It doesn't necessarily mean you're going to get breast cancer, it just increases your risk. And really this only accounts for maybe 10% of breast cancer patients. So only about 10% of breast cancer patients even carry this gene. The majority of people who have breast cancer, it's only these somatic mutations. So yes, again, you've probably heard of inherited forms of cancer, but the majority of cancers are only somatic mutations. They're not passed on to offspring. They're not inherited by offspring in any way.